Welcome to Biology Tutor. If you like this channel, please share and subscribe. Don't forget to click the bell button. First question. Today we are going to discuss about culture media. First question. Which of the following statements about selective culture media is correct? Statements. Selective media contain substances that inhibit the growth of certain microbes. They are primarily used to isolate fastidious organisms. Antibiotics are often added to these media for selective inhibition. Selective media are used in diagnostic microbiology to differentiate closely related species. Options A, B, C, D, which is correct. The answer is A, statements 1 and 3. Explanation. Selective media contain inhibitors like antibiotics to suppress the growth of unwanted organisms. They are used to isolate specific organisms, but not typically for fastidious species or species differentiation, which are usually the purpose of enriched and differential media. Second question. Which of the following media is particularly useful for isolating and identifying Mycobacterium tuberculosis? Statements. Lowenstein-Jensen medium, LJ medium is commonly used. The medium contains malachite green as an inhibitory agent. Egg-based media are preferred to support the lipid-rich cell wall. Charcoal is added to enhance growth, which is correct. Answer is B. Statement 1, 2 and 3. Explanation. LJ medium is egg-based and contains malachite green to inhibit contaminants. Charcoal is not typically used in media for Mycobacterium tuberculosis. Third question. Which of the following statements accurately describes blood agar? Statements. 1. It is a differential medium that allows observation of hemolytic activity. Second one, it is both selective and differential for fastidious organisms. Third, blood agar is enriched with sheep blood to support bacterial growth. It is useful for identifying hemolytic patterns of Streptococcus species. Which is the answer? Here answer is A. Statement 1, 3 and 4. Explanation. Blood agar is enriched but not selective. It allows observation of hemolysis alpha, beta and gamma and supports the growth of fastidious organisms like Streptococcus species. Fourth question. Which of the following culture techniques utilizes a low nutrient medium to promote biofilm formation? Statements. This technique is called a Calgary biofilm device. Nutrient restriction enhances biofilm formation as microbes adhere to surfaces. This technique is not suitable for high throughput testing. Biofilm growth on these media is observed with crystal violet staining. Option statement 1, 2, 4. Or all statements, which is correct. Here is A. Statements 1, 2 and 4. The Calgary biofilm device supports biofilm studies using a low nutrient medium. Crystal violet staining quantifies biofilm formation. However, it is designed for high throughput analysis. Fifth question. Which media is commonly used to isolate and identify Vibrio species? Statements. Thiosulfate TCBS agar is a selective medium for Vibrio. TCBS agar helps to differentiate Vibrio species based on colony color. The high pH of TCBS agar suppresses most non-Vibrio organisms. Bile salts and thiosulfate act as pH indicator. Which is the answer? Here answer is A. Statement 1 and 2, 1, 2 and 3. TCBS agar is selective for Vibrio species, differentiates them based on sucrose fermentation, and has an alkaline pH that inhibits other organisms. Bile salts and thiosulfate do not serve as pH indicators. Sixth question. Which of the following techniques is specifically associated with anaerobic culture media? Statements. Reducing agents like thioglycolate are added to the medium. Plates are incubated in a carbon dioxide rich environment. An anaerobic chamber or jar is required for culturing. Anaerobic indicators change color to confirm oxygen absence. Option A, B, C, D, which is correct. Their answer is option A. Statement 1, 3 and 4. Explanation. Reducing agents, example thioglycate and anaerobic chambers are common for culturing anaerobes. Carbon dioxide alone does not ensure an anaerobic environment. Seventh question. Which statements are true for Mcqs agar as a differential medium? Statements. It differentiates bacteria based on lactose fermentation. Lactose fermenters produce pink colonies due to acid production. Mcqs agar contains bile salts and crystal violet to inhibit gram-positive bacteria. It is specifically designed to isolate Neisseria species. Option statement 1, 2, 3, statement 1 and 3, all statements, statement 2, 3 and 4. Which is the answer? Here answer is A, statement 1, 2 and 3. Explanation. Mcqs agar is used for gram-negative bacteria and differentiates lactose fermenters. It is not selective for Neisseria species. Eighth question. Which statements are true for the technique of serial dilution and plating? Statements. It is used to estimate bacterial concentration in a sample. Serial dilution reduces cell concentration in a stepwise manner. This method is most useful for differentiating bacterial species based on morphology. Colonies are counted to calculate the original cell concentration. Options A, B, C and D, which is correct. Your answer is A, statement 1, 2 and 4. Explanation. Serial dilution and plating estimate the bacterial count by creating dilutions, plating them and counting colonies. Morphology differentiation is not the primary goal. Ninth question. Which of the following culture media is used specifically to promote fungal growth? Statements. SDA is formulated for fungi. SDA has a low pH that inhibits bacterial growth. SDA is enriched with chloramphenicol to selectively inhibit bacterial contaminants. SDA is used to isolate mycobacterium species. Which is correct here? Your answer is A, statement 1, 2 and 3. Savarot dextrose agar is formulated for fungi with a low pH and sometimes antibiotics like chloramphenicol or to inhibit bacteria. It is not used for mycobacterium. 10th question. Which of the following is true about the use of EMB agar? Statements. EMB agar is both selective and differential. It differentiates based on lactose fermentation. E. coli produces metallic green colonies due to acid production. EMB is used to cultivate anaerobic bacteria. Which is the correct answer here? Your answer is A, statement 1, 2 and 3. EMB agar is selective for gram-negative bacteria and differentiates lactose fermenters. E. coli typically produces a metallic green sheen and EMB is not suited for anaerobes. Thank you. If you like this channel, please share and subscribe. All the best for your exam.